Microfluidics is a field that studies the behavior of tiny droplets and fluids at the microscale, enabling precise control of chemical and biological processes. But what happens when these microdroplets are taken out of Earth's gravity? Microgravity presents unique challenges and opportunities for scientific research, particularly in the fields of space exploration, biotechnology, and material science. Understanding how fluids behave in low-gravity environments could open new possibilities for creating advanced materials, drug delivery systems, and even studying the origins of life. On Earth, gravity plays a crucial role in shaping how fluids move and interact. It causes droplets to settle, affects their shape, and drives convective mixing. In microgravity, however, these forces are nearly eliminated, meaning that surface tension and intermolecular forces become the dominant factors. This leads to unusual fluid behaviors that scientists can use to their advantage. For example, in the absence of gravity, microdroplets tend to remain perfectly spherical, which is useful for studying precise reactions that are otherwise disrupted by sedimentation or convection. One of the key applications of microdroplets and microgravity is in biomedical research. On Earth, cells and proteins in liquid cultures are often affected by gravity-driven effects, leading to uneven distributions. In space, researchers can study how cells grow and interact in ways that better mimic the natural environment inside the human body. This has led to experiments on the International Space Station where scientists have used microfluidic devices to study stem cell behavior, cancer growth, and protein crystallization with unprecedented accuracy. Could microgravity be the key to developing new drugs and medical treatments? Many pharmaceutical companies are interested in using space-based research to improve drug formulations. In microgravity, drug molecules crystallize more uniformly, which is critical for designing more effective medications. By controlling microdroplet behavior in space, scientists can optimize the formation of crystals used in vaccines and other drugs, potentially improving their efficiency when administered on Earth. Another fascinating area of study involves self-assembling materials. In microgravity, microdroplets can be used to create new types of structured materials that are difficult to form under normal conditions. For example, scientists have experimented with creating colloidal structures, microscopic building blocks that could be used in nanotechnology, coatings, and advanced electronics. By studying how these materials form and stabilize in space, researchers can design better materials for use in extreme environments, including deep space missions. How does microgravity help us understand the origins of life? Some researchers believe that early biochemical reactions leading to life could have occurred in microdroplets suspended in water-rich environments. In space, microfluidic experiments can simulate the conditions that might have existed on early Earth or other planets. By observing how complex organic molecules form and interact in microgravity, scientists may gain insight into the chemical processes that led to the first living cells. As space agencies and private companies push for human exploration of Mars and beyond, microfluidics will play a crucial role. Future space missions will rely on efficient water recycling, in-situ resource utilization, and advanced medical diagnostics, all of which depend on controlling microdroplets and microgravity. Could astronauts one day use microfluidic systems to print biological tissues or manufacture essential medicines directly in space? While microfluidics in microgravity is still an emerging field, its potential is vast. As researchers continue experimenting with fluid dynamics beyond Earth, we may discover entirely new ways to manipulate materials, improve medical treatments, and even answer fundamental questions about the universe. Could the future of scientific discovery lie not in massive machines or giant telescopes, but in tiny droplets floating in the weightless expanse of space? Microdroplets behave differently in microgravity due to the absence of gravitational forces, which normally dominate fluid dynamics on Earth. In low-gravity environments, surface tension and intermolecular forces become the primary factors dictating droplet movement and shape. Without gravity, droplets tend to remain spherical, as there is no force pulling them downward, reducing flattening or elongation effects. This unique behavior allows for precise control of droplet interactions, making microgravity an ideal environment for studying fluid mechanics. In space experiments, water droplets remain suspended in air rather than falling, allowing researchers to observe pure surface tension driven behavior. This is like a raindrop floating freely in the air instead of falling, revealing how its shape naturally forms under surface tension without external forces. Microfluidic devices in space operate using capillary action and surface tension instead of gravitational flow. Microdroplets in such systems can be precisely manipulated to perform chemical reactions, biological assays, or material synthesis without gravitational interference. The ability to confine and control small liquid volumes in space enhances labonic chip technologies for real-time analysis. These microfluidic platforms allow for efficient use of reagents, making them suitable for experiments in resource-limited environments like space stations. Astronauts have used microfluidic chips to test blood and detect contaminants in water aboard space stations. Microfluidic devices in space work like miniature science labs, performing complex reactions in tiny, controlled droplet environments. In microgravity, emulsions, mixtures of liquids that don't normally blend, behave differently, with reduced sedimentation and phase separation. Droplets remain suspended longer, allowing researchers to study the stability of emulsions without the influence of buoyancy forces. Droplet coalescence, the process by which smaller droplets merge into larger ones, is significantly altered in microgravity due to reduced external forces. This helps scientists investigate new ways to stabilize or control emulsions for applications in pharmaceuticals, food science, and cosmetics. Oil-water emulsions in space remain stable for extended periods, providing insights into how surfactants function in low-gravity environments. This is like shaking a bottle of salad dressing and watching it stay mixed longer than expected without the oil and vinegar separating quickly. Microgravity provides an optimal environment for protein crystallization, a process crucial for developing pharmaceuticals and studying biological structures. On Earth, sedimentation and convection can distort crystal growth, while in space, droplet-based crystallization is more uniform and controlled. Using microfluidic droplets in microgravity enhances crystal purity and size, leading to better drug formulations and biomedical applications. These insights are critical for designing targeted therapies, vaccines, and enzyme-based treatments. Space-grown protein crystals from the International Space Station (ISS) have helped researchers improve drug formulations for conditions like Parkinson's disease and cancer. This process is similar to growing salt crystals slowly in a still solution rather than being disturbed by external movements. Microgravity provides an ideal environment for studying chemical reactions without sedimentation or convection effects, which can influence reaction rates on Earth. Using microfluidic droplet systems in space allows for precise control over reaction kinetics, phase separations, and mixing efficiency. Researchers can design novel self-assembling materials, nanoparticle synthesis, and controlled reaction pathways using space-based microdroplets. These advances have potential applications in pharmaceutical synthesis, nanotechnology, and materials science. Scientists have conducted nanoparticle formation experiments in space to observe how materials self-assemble in weightlessness. This is like mixing ingredients in a perfectly still environment, ensuring only molecular forces determine the reaction outcome rather than external disturbances. Microdroplet-based microfluidics enable cell cultures to grow more naturally in microgravity, avoiding issues like uneven growth and mechanical stress from gravity. Space-based microfluidic platforms allow researchers to study stem cell differentiation, organoid formation, and tissue development under unique conditions. These findings could advance regenerative medicine, cancer research, and drug testing, leading to breakthroughs in personalized medicine. The ability to control microenvironments within droplets allows for precise manipulation of cell behavior in space. 3D tissue cultures developed in space exhibit more realistic structures than those grown under Earth's gravity. This is like allowing a plant to grow without external constraints, letting it develop freely in all directions. Microfluidic droplet systems in space are being tested for advanced water purification, filtration, and oxygen generation in spacecraft. Without gravity, traditional filtration methods become inefficient, requiring novel droplet-based separation techniques. Microfluidic technology allows for membrane-less separation of contaminants, pH adjustments, and nutrient recycling, making closed-loop life support systems more efficient. These technologies could enable long-duration space missions by improving water recycling and environmental control. NASA has developed microfluidic devices that process astronaut wastewater into drinkable water with minimal energy input. This is like creating a self-sustaining aquarium where water is continuously filtered and recycled without external waste buildup. In microgravity, fuel and cryogenic propellants must be managed with non-gravity dependent methods such as capillary-driven microfluidics. Droplet-based fuel handling can improve mixing efficiency, precision in dispensing, and controlled fuel combustion for space propulsion. Researchers are studying how liquid droplets behave in microgravity to optimize fuel transfer and storage for interplanetary travel. These systems could lead to more efficient and safer fueling methods for future spacecraft. Experiments on fuel droplet combustion in space help refine spacecraft engine designs and fuel efficiency. This is like using precise droplet injections in a car engine, ensuring optimal combustion and minimal waste. Microfluidics holds promise for advanced manufacturing, autonomous chemical synthesis, and fundamental physics research in space. Ongoing research aims to develop self-regulating microfluidic platforms that require minimal human intervention for long-duration space missions. These technologies could also benefit Earth applications such as pharmaceutical synthesis, food production, and environmental monitoring. Future missions may integrate AI-driven microfluidic experiments where autonomous systems optimize processes in real time. Upcoming space missions plan to use automated microfluidic reactors to produce biomaterials in space. This is like having an autonomous kitchen that prepares perfect meals by precisely mixing ingredients in